A very good evening to everybody. Welcome to this Retina UK information webinar. This is one of a series of webinars we're hosting, and we'll be delivering at least one on a different topic each month. We are really pleased to have with us today Dr. Salwah Rehman. Salwah is a clinical research fellow at Oxford University Hospitals. She's currently working on the ongoing gene therapy trials for choroideremia and RPGR X-linked retinitis pigmentosa under the supervision of Professor Robert MacLaren. We are also joined with us this evening by Kate Arkle, our Research Development Manager, who will be collecting questions throughout the presentation this evening. So there are a couple of ways for you to ask questions. You can either type them in the Q&A section at the bottom of your screens. These questions will be asked by the team on your behalf. Alternatively, you can raise your hand by pressing the Alt and Y key if you're using a Windows computer, or the Option and Y if you're on a Mac. If you're on a tablet, you'll find them under Reactions. We will then, as we say, we'll ask you to unmute your microphone and you can ask the question yourself, and those will be done at the end of the presentation. So please, if you have any questions as Sal was going through, leave them or raise your hand and we will do a Q&A session at the end. We will endeavour to answer as many questions as we can. However, any questions we're not able to get to today will be followed up over the coming weeks. So thank you again for joining us, and without further ado, I'm delighted to introduce Salwah.
Thank you, Matthew. I'll just share my screen. So thank you, everyone, Matthew and everyone at the Retina UK for having me deliver this talk today. And thank you to everyone who's joining in tonight. As Matthew introduced, I'm Salwah Rehman, and this is my second year as a clinical research fellow under the supervision of Professor Robert MacLaren at the Oxford Eye Hospital. A little about my background. So I completed my medical training in the University of Liverpool. And while I was there, I took a year out to study a master's in research. And more specifically, one of my projects involved looking at a novel way of measuring eye pressures. So that was my kind of first introduction into the research world and got me really interested in ophthalmology. After medical school, I then completed my junior doctor training in Bolton Hospital in Greater Manchester. And then after this, I wanted to take some time out to explore sort of my interests. And I sought out this position in August 2020. And it gave me a really fantastic opportunity to learn about, you know, this exciting field of gene therapy, which was completely new to me, as well as get more experience into the clinical trials and ophthalmology. So today I'll share with you some of what I've learned over the past year and a half, and please feel free to ask questions at the end. Okay, so this is what I'll, just to set the scene a little bit, I'll first briefly talk about what part the clinical research plays in the process of developing new drugs. And then I'll explain a little bit about what we mean when we talk about a clinical study, before talking in more detail about the different phases of clinical trials. I'll then go through what a study plan or a study protocol includes, who's involved in clinical trials, and some of the things you should consider as potential research trial or clinical trial participants or patients. After that, I'll give a brief update on the current and recent trials I've been a part of since being in Oxford. And finally, how you can register your interest should you be interested in clinical trials. So firstly, I mean, we're all here one way or another because we want new treatments to become available for conditions that are yet untreatable. So there are 5 main steps in the drug development process. So typically, this starts with the discovery and development of a new drug. Researchers may learn new insights into a disease process and design a product to stop or reverse the effect of the disease. And this steps usually begins in the lab with many tests on hundreds of molecular compounds, from which only a small number show promise. And so further experiments would be done to gather more information on how the compound is absorbed, how it's distributed, metabolized, and excreted, its potential benefits and side effects, the best dosage, best way to give the drug, and how it interacts with other drugs and more. So following this, the next stage is testing this new drug for potential to cause serious harm or damage. The two types of preclinical studies are in vivo or in vitro. So in vitro being in the lab and in vivo being animal testing. So usually preclinical studies are not very large. They provide important information on dosing and toxicity. And while these preclinical studies answer really important questions about the safety, they can't tell us how the drug will interact with the human body. And this is where the next stage in the process comes in, which is the clinical research involving human participants. And this involves testing for safety and effectivity of the product, of the treatment or intervention. And we'll talk a bit more in detail about clinical trials in just a moment, but just for the sake of a completion, once a trial has been completed and reviewed by the Food and Drug Administration, the FDA, and they have decided to approve or not, the next step is basically, so following the review, the final step is post-market safety monitoring. Because although we have all this information to approve the drug, we don't have the long-term kind of data on the drug, and the true picture of the drug safety sort of evolves over the months and years in the marketplace and when it's being offered to patients. So an example of this kind of monitoring after the approval is the PERCEIVE study, which is gathering data for patients who have been treated with Luxturna, you may have heard of. And this is a new gene therapy treatment for RPE65. So currently patients are being treated in Oxford on Luxturna, and we are gathering data on the PERCEIVE study for that. So this is an example of post-market safety monitoring. So now going back to what is a clinical study? So there's many different types of research studies involving human participants, but the two main types of clinical study are clinical trials and observational studies. And the main types of the clinical studies, so in a clinical trial, participants receive specific interventions according to the research plan or protocol, which is created by the investigators. So these interventions may be new medicinal products like drugs or devices or procedures. Some trials may compare a drug to a new drug, compare a new drug to one that already exists, or compare it to a placebo, which contains no active ingredient or to no intervention at all. So when a new drug or a new approach is being studied, it's not really known whether it will be helpful or harmful or no different to what's already on the market or what's already available, including not doing anything. So this is why clinical trials are needed to determine the safety and efficacy of a new intervention by measuring certain outcomes in the participants. So for example, giving a new drug to participants with high blood pressure and testing them to see whether that drug is able to lower their blood pressure, or testing a new gene therapy treatment in RP patients, the retinitis pigmentosa, to see if that treatment is able to improve the patient's vision or slow down the degeneration in those patients. So there's all these different types of interventional trials depending on what you're testing for. And these clinical trials are used to develop new drugs, and they occur in phases which are defined by the Food and Drug Administration. So these are the phases of the clinical trial. So we've discussed sort of basic research and preclinical research before a clinical trial can move on to sort of the different phases. To do this, to move a trial beyond the preclinical stage, the sponsors have to begin what we call the investigational new drug process application. And this is a lengthy kind of process, which an application that before beginning clinical research includes all the details about all the prior animal testing that's been done, all the data to include safety and toxicity so far, how the drug's manufactured, what the study plan is, any data from any prior human research, and any information about the investigators. And then we move on, once that is approved, to the first phase, which is the physiologic or toxicity and dose finding study. This usually consists of a few volunteers, about 20 to 100 people with the disease, and the main goal is to show possible side effects and the safe range of a possible new drug. And this part of the study can usually take several months to a couple of years. In the second phase, looks at the drug efficacy and side effects. Efficacy being, is the drug having the desired effect? Is it slowing down degeneration? Is it stimulating new cell growth, etc. While also at the same time monitoring for any adverse events or side effects. And this phase can again take a couple of years, and several hundred people are needed to fulfill this goal at this stage. So the final phase before approval, the phase 3, tests the efficacy, the effectiveness, and safety of the drug in many more thousands of volunteers and different places and centres. And this allows us to see how the intervention translates to practice. Can it be delivered safely in a variety of different centres and safely and effectively? And it compares the intervention to the other available treatments already out there, if there are any, and this data is collected. Again, this phase can take a number of years, so up to 4 years or even more, with many thousands of volunteers, and that is if everything goes smoothly. And finally, phase 4 trials, so after approval of a drug by the FDA. This is after the drug's already been licensed, and they aim to evaluate the treatment over an increased duration for post-marketing and surveillance. So they aim to find out more about the side effects and safety of the drug, what the long-term risks and benefits are, and how well the drug works when it's more widely used. So this slide, basically the aim is to give you an overview of sort of the different phases and the timeline of how long each phase takes when everything goes smoothly. So one example I have, which I'll share with you, and which put this kind of time scale into perspective for me, was the Reneuron trial. This is the stem cell research trial, stem cell transplantation trial for patients with retinal degeneration, which Professor MacLaren and his team tested on mouse models about 15 years ago. And from that preclinical stage to the first in-human phase 1 trial, it took 15 years to get there. So I've basically contradicted everything I said just now. But that's just to illustrate that, you know, from preclinical stage to actually treating the first patient at phase 1, that's taken us 15 years. And we treated our first patient in Oxford 3 months ago. So that was a big, big moment for the team and for, you know, for this community, I think. So the timescales are not always predictable and straightforward because for many of these diseases, there's still so much more that we have to learn. But we're very optimistic for the future because there's some amazing, amazing work being done in the lab. So just moving on to, I mentioned earlier that each study at each phase needs a detailed study plan or a study protocol. So what does this exactly include? So before a clinical trial begins, all the prior information on the drug is reviewed to develop research questions and objectives. The study protocol is then written to set the inclusion and exclusion criteria, the selection criteria, the study size, the length of the study, what controls will be used, the mode of delivery of the intervention or drug, the dosages that will be used, any tests and assessments that will be conducted, and how they'll be conducted and at what time points they'll be done, and how the data will be analyzed at the end. And I mentioned controls there, and that's a very important aspect. I think the eye is a great organ to test gene therapy because we have 2 of the same organ in our body. So one of them acts as a natural control to compare the effects of a treatment versus no intervention. And so this is a great way to eliminate any bias in the study. This is a very busy slide, but essentially it's just to highlight that there are very many experts in a variety of different fields involved in the design and delivery of a trial. And just to mention that all the people involved have to be trained in good practices so that we can basically deliver a good trial. So what is it that you should consider as a participant or as a patient? What's important for you? So if you were to come into clinic and you were considering joining a clinical trial and there was something that we could offer you, we would obviously talk about the trial, give you all the information. All the information would be discussed with you verbally in clinic, as well as giving you a participant information sheet. Which will include all the information you need about the risks and benefits of the treatment, the eligibility criteria, the inclusion and exclusion criteria. The process is sort of discussed with you, and we get an informed consent from you. So you get all the information, all the risks and benefits, and are offered the chance to kind of relay the information back to us and ask any questions or any misunderstandings. So we make sure that you're very, very clear of all the information necessary to make this decision, and we get a written consent from you. That being said, it's not, you know, a signed informed consent isn't, you know, a contract. So participants can withdraw from a study at any time, even if the study is not over yet. So it's just to get your permission to join the trial, but you can leave at any time should you wish not to continue. The commitment that it takes, because it is a huge commitment to kind of sign up to a trial, and we always are very frank and open about this because everyone has a lot of things going on in our lives. We're busy with work, with family. And so, you know, taking time out for these very frequent clinic visits is a commitment, and also a commitment to do these tests and assessments each time you come. So that's a big consideration once you join the trial or in the initial stages. You go through a screening process, and through the screening process, you do all the kind of assessments and tests that are needed to determine whether you're eligible for the trial, and whether you meet the criteria, and whether you are able to conduct the tests reliably each time, which is very important for us to sort of monitor your progress and monitor any benefits of the therapy that you've received. So the screening process can occur in a number of visits, so 2 to 3 visits usually, following which you then have your treatment, and then after that there's usually visits that go from once a week to sort of day 1, day 8, month 1, month 2, month 4, month 6, up to sort of a year, and then following that they sort of increase in the duration of follow-ups. So like 6 monthly follow-ups usually is what we do for the long-term follow-ups for many of the trials that we have going on at the moment. But it is trial specific, and any of this information would be sort of made very clear in the participant information sheet, and the kind of commitment that the trial demands from you, basically. Just a few examples of the sort of tests that you would be required to do should you sort of join any trials. So this is an Natdrs chart, which is basically a visual acuity chart. So you're required to sort of read that to check your level of vision each time you visit, and that helps us to monitor how your central vision is doing. These are images of scans of the back of the eye. So the long one in the back here, this is the cross section of the retina, and this one is sort of an image of the back of the eye if you're looking from directly in front. So this is the patient's right eye, and here through this green line we're looking at this sort of cross section of the retina with the different layers of the retina showing, and we can see this bit here is the normal sort of macula, or the central part of the retina. But as you go out into the periphery, the retinal layers are thinned out, and essentially this patient has a retinitis pigmentosa as a result of an USH2A mutation, which causes this sort of appearance. And this image at the bottom here is an autofluorescence image, again, of the back of the eye. Here is the optic nerve with the vessels, and this sort of pigmentation that you can see in the periphery, that's the kind of a hallmark of retinitis pigmentosa. And this in the middle, this sort of ring appearance, is where we can see these structures here. And this is a macula, so the very central part of your vision where you have the cone cells. And between this bit and this bit, all of this central bit is the normal, healthy, functioning retina, which is this bit. And as you move further outwards, which is towards this kind of white ring, you see these cells have lost part of their segments, which is the outer segments, and this sort of gives the appearance of the ring, which is basically it's termed ciliopathy, where you lose it, there's like a transition zone where you lose the outer segments, and this kind of pattern of degeneration is typically seen in an Usher 2A retinitis pigmentosa. So it's very important for us to sort of get these scans at each visit and monitor your progression from each visit and from before the surgery or before the therapy to after the therapy. This is just to illustrate sort of a color picture that you would have done when you come to clinic. It's just a nice picture to look at. And here's a picture. So this is a visual field test that you would have called microperimetry. And essentially this is the result that we would get and interpret. So this is from a patient with choroideremia, and as you can see here, this is the choroideremia island, typically seen in this condition. So this bit of the retina is functioning well. And so when you do the visual field test, it measures points of sensitivity. You basically focus on a red light and click the button each time you see the light blink in these in this sort of pattern in this area, and it looks at that kind of central vision and picks up on the areas of your retina which are sensitive, and we sort of monitor that over your follow-up visits to see how the disease is progressing, whether you've had some kind of gain in your sensitivity in your retina following treatment, things like that. And also it kind of tells you about your fixation and basically how you're progressing. So I thought I would give a little update on the current and recent trials that I've been a part of since being in Oxford. So for choroideremia, GT1 was a phase 1 trial for choroideremia. This was before my time, but this was I included that because it's the very first phase. We then had the NIGHT study, which is the observational study. So patients enrolled onto that who didn't receive an intervention, but we monitored them over a period of time to see how the condition progressed. The REGENERATE was the next phase, the phase 2 trial. This closed earlier in 2021, and the last patient was treated in June 2019. And then the STAR trial came after that, which is a phase 3. So all these phases testing these kind of safety and tolerability and efficacy of the drug. So for the phase 3 trial, patients were treated sort of around November, December of 2020. So that's now not recruiting anymore. So the results for the STAR trials were released earlier on from Biogen, which basically didn't meet the official sort of finding was that it didn't meet the primary endpoint. So currently the trial has been suspended, but you know the labs are not making the vectors at the moment because they're all busy with the COVID vaccine. So at the moment the trial isn't moving forward, it's been suspended, but we are hopeful and we'll watch this space essentially, and soon as soon as we hear any information about that trial moving forward, we will let everyone know. For all of these trials, the patients are being followed up long term on the Solstice trial. So this is a 5-year follow-up of all the patients enrolled on the STAR and REGENERATE trials. So currently, I am still seeing patients on the long-term follow-up. Over on the other side, we have the retinitis pigmentosa trial. So the Zolaris was the observational study. Sirius phase 1 and 2. Basically, that trial is also finished. The last patient was treated on the 30th of September in 2019. And currently, patients from this study is being followed up on the long-term study, the Solstice, up to 5 years. And then just below that is the Reneuron phase 1/2 trial, which I mentioned earlier on, which is currently we are recruiting patients for. So far we've treated 2 patients in Oxford with this trial. So I think we have the go-ahead for the new year to kind of recruit further patients onto that. And then finally, at the bottom, Perceive is the trial we're collecting data on patients treated with Luxturna, which is sort of collecting the long-term data. And finally, how you can register your interest. So if you go on the iResearch Oxford website, you can fill in kind of the contact us form there, or alternatively, you can email the Ergo email address, so ergo@oeh.nhs.uk, and or email the office on that number there at the bottom. And we're always very happy to hear from you. And just a little thank you to everyone in the team. So this is our lab group. This picture is from our recent annual lab dinner. We get dressed up once a year, which is very nice. And we have lovely socials at some other times in the year, which is, you know, we have some fun. So thank you very much for listening to me today. I'm sure there's questions and I'm happy to answer.
Thank you ever so much, Salwah. That was really interesting. Or just probably best if you stop sharing and then, yeah, perfect. Yeah, I think it's really important for our community, because there's so much coming through now. There's such a lot going on, and it's getting really quite exciting. And I think we're really grateful to you for giving us an overview of how the process works, because I think people need to understand that so that they can make really informed choices about whether these trials are something that they perhaps want to take part in. And I think it's helpful also just to know what treatments have been through, even once they get to sort of the NHS and wider availability. I think it's reassuring to understand how they've been tested. I always forget that whole investigational new drug bit of the process that people have to go through, all the sort of bureaucratic hoops that have to happen just to move from your preclinical work into human studies. And yeah, that obviously exists for safety reasons, but it's always just worth us remembering how much process there is behind all of this and why it sometimes takes a bit longer than
I really wanted to highlight just exactly how, you know, how much it takes at each step. And yes, it's disappointing to hear sometimes when trials come to a hold, or, you know, things aren't moving as fast as we would like to. But I have a newfound kind of appreciation for these things coming into this only sort of over a year and a half ago. Before this, I had the same kind of ideas about clinical trials, you know, but now being on this side and looking at really how the process works, there's a lot of work and a lot of people involved, and a lot of really good research work happening in the background. So I think it's we're all really optimistic and hopeful that lots of good stuff is going to come out in the future.
It actually ties in with one of the questions that we've got in the box, if I can just move it up. So Matthew said, do you think trials will begin adopting the way that the COVID vaccine was developed, with phases overlapping, or do you think that was a one-off and unique to the pandemic? This is something that people are asking quite a lot at the moment. You know, if the COVID vaccine could happen so quickly, why can't everything? But I guess
I think I mean, I'll try my. That's a very interesting question. Actually, it's making me think. I think with conditions like these inherited retinal diseases, they progress at varying rates and at slow kind of rates of progression. So oftentimes you need that time to see if an effect can be found after a treatment's been administered. In my experience, for example, take into account the STAR trial, phase 3. The primary endpoint for that was, reading 3 lines on the chart. So were patients able to read 3 lines on the chart at the end of the trial? And some patients were, but there wasn't a good enough, or many people, or not enough patients were able to meet that criteria. But if you look at the secondary outcome, which is reading 2 lines, many patients were able to meet that criteria. And I just wonder whether if the trial was to go on for a bit longer, we may see that over time more patients would have a more meaningful outcome. Mm-hmm. Or it could be that the outcome should have been 2 lines, and that's the sort of the mark that we should be aiming for. So I don't know, I think in my mind I feel like there needs to be a long enough time period to see whether an effect can be found in these conditions. But it does make you wonder how they can do it so quickly for the COVID vaccines.
Yeah, I think you make a really, really good point, and it's really important that we don't rush these things and actually miss a potential beneficial effect. Absolutely. And I know that some of our community would say, it's interesting actually the way you describe that endpoint, because I hadn't thought of it like that you sort of set this endpoint of a very black and white binary, you can or cannot read these lines. And so many of our community say any vision is better than no vision, and even if I could just stop it where it is, and yeah, that doesn't quite seem to marry up. So that was an interesting aside, if you like. But I guess as well, perhaps the COVID vaccines, we have so much vaccine background and knowledge, and whereas gene therapies and things like that are so new,
relatively new. Yeah.
And I wondered as well, it was interesting that you mentioned the vector production actually stopping because everybody's churning out COVID vaccine, and I hadn't thought of that. So has the COVID vaccine development, or if you like, and that concentration actually slowed other things down a little bit? Has that had much
Absolutely.
impact?
I think all the labs are making COVID vaccines, so there is not enough vector to go around. So there was definitely trials that slowed down further kind of stages. For example, for the RP study, the next stage hasn't really been announced yet. We haven't heard anything about that trial moving forward, and I suspect part of the reason for that is actually the resources. Is there enough vector being produced to do that?
Yeah. So just to outline for those that don't know, vector is the viral package, the virus package that we put gene therapy, the genetic material inside to carry it into the back of the eye, the cells at the back of the eye. But obviously some of the COVID vaccines use modified virus to produce the immune response we want from a vaccine. Is that correct, Salwah? Yeah. So that's why vector manufacturing, it's a very specialist manufacture process, and that's why those places that can make that vector are a bit preoccupied at the moment. Okay, we'll move on to some other questions. Paul says, do you feel that older patients, perhaps in their 60s, are a lower priority for gene therapy? Now, I don't know whether Paul means for a clinical trial necessarily or for the actual treatment, but I'm guessing that the main issue is not age per se, but the degree of deterioration of the retina.
Yeah, I mean, there's different criteria that play into the selection process, and these criteria are very clearly laid out for us before we begin recruiting. Part of the criteria is, does the patient have the required gene mutation that we're looking to study? Does the patient have the level of vision that we want to treat? So we don't want to treat patients with too good vision because, again, what's the risk versus benefit for those patients? We don't want to treat patients with too poor vision because with that poor vision, are they able to reliably and consistently repeat the tests required for us to make, a measurement or an outcome to see whether the treatment is working or not? So these criteria are laid out for us before we begin recruiting. Age doesn't really play a part into it. It depends on your sort of general health, whether you can take or tolerate I'm talking about gene therapy trials here. So, a general anesthetic, for example, and how's your mental and physical health, basically. So no, age is not a discriminatory factor. Obviously, some trials state that the patient needs to be above 18 if it is a trial recruiting adults. But no, I haven't come across, an exclusion criteria based on age.
And gene therapy, certainly from what I've heard, seems to be more likely to work when things haven't progressed too far, because you need surviving retinal cells to be able to make use of this treatment. But again, that's presumably even once the treatment is available. For example, Luxturna, obviously you've got to have the correct genetic diagnosis, but then the decision is going to be based on the actual state of your retina rather than your age. So it's about that rather than, Okay, lots of questions. Oh, hang on. Okay, so Catherine says, can any patient from across the UK get in touch with you in Oxford? Are there other sites offering recruitment in different areas of the UK? I guess that's going to depend on the trial to an extent, but
yes, I think anyone is very welcome to get in touch if you just like some information about the trials that we're doing here specifically, or if you would like to be seen by one of the clinicians in Oxford, you're very welcome to get in touch. You can also get all the information about the trials that are happening in Oxford on the iResearch group specifically, but I think for specific trials that aren't happening in Oxford, you may be able to contact the other centres directly.
So Catherine, it depends on the trial as to what centres are taking part. The Reneuron trial, for example, as far as I'm aware, Oxford is the only UK centre involved in that trial, and you're just recruiting a very small number of participants for that at the moment. Moorfields sometimes recruits patients for some trials. There's other places around the country that might do as well. Somebody else has actually said, what's the website for those wanting to register an interest in volunteering for a study? So could you just remind us of which the Oxford one is, and we can perhaps also get some of this information sent out as a follow-up, possibly to the webinar.
Yeah, I can forward you those details. The website is iresearchoxford.org.uk.
Perfect. And yes, I know that specifically to Catherine's question, because I'm sometimes in touch with the clinical trials coordinators at Oxford, and they're very happy for anybody anywhere to fill in a form and give Oxford permission to access your medical records, and then potentially get in touch with you if they think that you might be suitable for a clinical trial. But it is worth remembering, as Salwah mentioned, there are always a lot of criteria, and that sort of, I guess you're able to get a certain amount of idea about people's eligibility from their medical records and their history.
Yeah, absolutely. So the things that are very useful when you do get in touch with us and are interested in trials, excuse me, are so your visual acuity. So if you could get that from your local ophthalmologist or opticians, And that's and any genetic confirmation is very, very helpful, because for many of the trials, they have specific genes that are being treated or may be eligible. You don't want to recruit patients with gene mutations that may have other trials that may be beneficial for us, for example. So for Reneuron specifically, we are looking for patients who have a cone rod dystrophy, retinitis, ideally, a confirmed retinitis pigmentosa mutation, but it doesn't necessarily have to be. So if you have a cone rod dystrophy, and then we basically base it on the strict sort of visual acuity criteria that we have. So it's very helpful to have that information, and we can basically take that to give you further information about your eligibility.
Okay, but then obviously the screening still has to happen, and that involves a huge number of tests and things, doesn't it?
And we do have, we have to remember that you can go through to the screening process, but then you could fail the screening, and which is very common. Some patients may have the right gene, they may have the right kind of visual acuity, but then they may not have the kind of consistency or the ability to do the testing that's required. And so unfortunately, in those circumstances, you can't move forward, because essentially you want to recruit patients who can do all these things so that we can get the best data, so that we can move the drug development process forward.
Yeah, I guess not being able to take part in the trial, aside from the fact that obviously the trial takes time and the condition perhaps continues to progress, but not being able to take part in the trial doesn't necessarily mean that you couldn't have the treatment if it were to later become available. Yeah. It's just that the trial demands very rigorous kind of measurement and standardization. Would that be fair to say?
Yeah, definitely. Yeah. So, it's so that we can get the best quality data, so that to move the trial forward, because we all want to get to the next stage and get the drug approved, right? So, the better the data is, the better the patients we recruit, the quicker we can move on to those stages, and then the treatment can become available for everyone. It's just that at different stages, the inclusion criteria requires vision of a certain level. So you don't want to start with patients with really good vision, because again, it comes down to the risk benefit of it.
Yeah, so actually those people with more advanced vision loss are useful to you. I hate to put it like that, but in the very early stages, because
people of all vision are very useful. But yeah, at certain stages, we're looking for certain levels of vision.
Yeah, okay. I think some specific questions about certain trials. Somebody's asked, are there any trials for the EYS gene in RP? Now, I don't know off the top of my head, that is ringing a bell, but I don't think it's a clinical trials I've looked up for that. I think somebody's asked me about that before. Are you aware of anything with EYS?
I can't guess off the top of my head, so I'm sorry.
That's fine. I can follow up with that. I can have a look online and follow up with that. Matt, if you could make a note of that question, or it will be recorded anyway. Sorry, I'm just having some problems scrolling on my laptop at the moment. Debbie says, can members of the public access the results from trials, previous trials or current trials? Kind of partly commercially sensitive, aren't they sometimes? But
so the information that's publicly available, so on the sponsor's website, for example, the two I can think of is the recent STAR trials kind of press release, and the Sirius phase 2. The results from these are available on Biogen's website. Now the research manuscripts, which may be a little bit too advanced for everyone, they offer more in-depth results for these studies. And some of these have been published, and some of these are currently in the works at the moment. So I think usually patients do get an update when more information about the study results are available.
Somebody's actually asked a specific question about results from the Sirius phase 1/2 trial. Has that data been fully analysed yet? Is that?
I think they are working on it at the moment, so it's not available publicly yet. It's still in the works. It's coming.
Do you know how long, on average, that brings me to ask, that data analysis takes? Because I think that's important as well, that we all have to remember that that's another job that has to be done, that's going on behind the scenes. So yes. From a trial ending to that data being available, do you know how long that normally is?
Again, it's kind of like when I spoke about the timelines before, they can take a long time. You know, I think the Sirius phase 2 ended in, when was it, in 2019, the last patient in Oxford. It takes quite a bit of time because it involves so many different people of different specialties and experts in different fields. But, you know, rest assured that this data is not going to go to waste. It is being analysed, and we just have to sort of watch the space. I can't give you an exact time. No, unfortunately.
I know that when you're living with sight loss, it can feel just such a laborious process, and I hope it's maybe a tiny bit of comfort to remember that people are working really hard on it, and there's reasons why it all takes so long, and ultimately we want to get the best results at the end of it for everybody going forward.
Yes, things have slowed down a little bit because of COVID, but things are running up and back up and running again.
Jenny says, how do we find out which genetic mutation? I'm not quite sure, Jenny, whether you mean how do you find out which genetic mutation you have, or what clinical trials are going on. So perhaps between us we can try and address both of those two questions. So Salwah, do you want to just explain to people how they would go about finding out which genetic mutation they had themselves?
Yeah, so if you're being seen at your local ophthalmologist or local hospital or local eye hospital, you can request a genetic test to be done. And that basically just requires some bloods to be taken from you and your permission to run a genetic test on that, to test for the genes that are suspected to be, you know, causing the disease. So if you're, for example, suspected to have RP, or you have a clinical diagnosis of RP, then you can get your genetic testing done to look at those specific genes. And having that report is so important when you come to see us, because it gives us a lot of information and a confirmation on the diagnosis.
Okay, and Jenny, we have got some information on our Unlock Genetics website. If you're ever struggling to get a referral for that, we've got some information about that, which is at retinauk.org.uk/genetics, about how to get referred. But the other possible, I think Jenny possibly also meant, how do I know what trials are going on for what genes? My go-to would be clinicaltrials.gov in the first instance. Have you got anywhere else that you can recommend, Salwah?
To be honest, that's the one we recommend in clinic, because they have, you know, it's required for any ongoing trials to be listed on there. So chances are you will find any trial on there.
Okay,
so
that's a website which is www.clinicaltrials.gov. It's actually an American government website, I think, but people all around the world, companies and researchers all around the world can register their study on that website, and you can search it. So if you want to search for retinitis pigmentosa, and then potentially you can add in another term, and you could put in there the name of a gene, and that should find, if there are any studies for that particular gene, they should pop up. But I always have to caveat clinicaltrials.gov with the fact that anybody can register any study on there. So it's always worth, if you're going to get in touch with a study and think about taking part, that you just run it past your own ophthalmologist or another expert, just to check if it's not very obvious that it's not bona fide. Obviously, if it's taking part at Oxford or Moorfields or somewhere like that, then it's highly likely that it's a proper study. Okay, I think, oh, hang on. Paul just wanted to say thank you. You're very welcome, Paul.
You're welcome.
Oh, Jenny, just quickly, I think we've got 2 minutes. What's the most common genetic mutation in RP? And if there's one with large numbers of people, are the trials geared towards that? My son's genetic mutation is FAM161A. I haven't heard of that one. Are there any trials on that? It's not necessarily how many people, is it, that there are living with a genetic mutation that dictates what gets studied or what gets trialed?
Not as far as I know, but it does help to have, the data. So that's not to say that rare genes are not being looked at, but obviously the more common genes, like RPGR, are already in trial phases. So that kind of leads you to believe that the rare genes are overlooked. But essentially, we do need the data, the background data, for it to push forward, because it's commercial at the end of the day. You have to remember that although we want to, create the treatments for all of these, the sponsors have their own sort of commercial interests that they look at too.
I know it doesn't mean that very rare genes are a challenge, but there are sometimes other ways of addressing those problems. And Robin Alley, who was at UCL and is now at King's, has done some work with a very rare mutation where they've just done it on a special license at Great Ormond Street. So sometimes there are ways around these things, but as Salwah says, it obviously is.
There are rare genes that we are looking at in our own lab group. So there is, even though they may not be in clinical trial process or stage right now, there's preclinical research going on in those rare genes. So the future's bright.
Yeah, absolutely. And of course, there's treatments that ultimately won't
will kind of push for the rarer ones to have treatments developed for as well.
Yeah. Okay, thank you very much, Salwah. I think that brings us to quite a nice conclusion. Thank you so much for your time this evening. Thank you, everybody, for joining us. Matt, do you just want to follow up briefly with how the recording is going to be dealt with and things?
Yeah, absolutely. Huge thank you, Salwah. That was a fantastic presentation. I've certainly learned a lot.
Thank you for having me.
And of course, thank you to everyone that's joined us for our webinar today. As mentioned at the beginning of the evening, we are planning at least one of these webinars every month going forward, and the details of the ones for 2022 will be available shortly. Just a gentle reminder that Retina UK is a registered charity. We receive no government funding and rely on our wonderful supporters, such as yourselves, to raise the funds needed to provide vital services and invest in groundbreaking medical research, some of which obviously goes towards these clinical trials that we've heard about today. So there's many ways you can kind of get involved with this. And as Christmas is literally just around the corner, there are a number of ways you can get involved and help us raise some of these funds. So we have got some Christmas cards available for purchase. If you're feeling a bit energetic, there's a virtual Santa Dash that you can get involved in. And please save any stamps from your Christmas cards and send them into our stamp appeal. You can find out more on the website www.retinauk.org.uk/Christmas, or you can call the office team on 01280 821 334. So just to let you know, after this session this evening, we will be sending out an email. This will be with you over the next couple of days, which will have details of where you can re-watch this evening's presentation. and details of how you can book onto any future webinars that we hold. We'll also be looking for your feedback. Feedback is a gift, and we take on board everything that everybody says, and it really does help to shape these webinars that we deliver and our other services. So please do fill in the feedback form and send it back to us. So once again, thank you to Salwah, and thank you to everybody who joined us this evening. Thank you very much. Goodbye.